Hi everyone. Yeah, thank you so much, Trevor, for the invite to talk today. I'm going to be giving you sort of a high-level overview of some of the work that we're doing at H3D, and you'll see that we've managed to build some really nice biology capabilities in-house, but I think I'll also be able to convince you that we're still lacking some. And so just, yeah, how we could use structural biology going forward. Okay, so for those of you who don't know, H3D is a drug discovery center embedded in the University of Cape Town. It was founded in 2010 by Professor Kelly Chibali, who is a synthetic chemist by training. Um, and really, the mission is to develop new medicines for infectious diseases. But coupled to that, we're also looking at building Africa-specific models for improving treatment outcomes in African patients. Coupled with doing drug discovery in Africa is the need to develop platform technologies to support this. And then really crucial for sustainability is training African scientists in drug discovery related sciences. So just briefly, we have a range of programs at H3D, which are funded by both local and international sources. We work on three disease areas. So malaria was really just the first main one in our portfolio, and this is where we really had a early success on it with a phenotypic based drug discovery program. So at that time, it was all driven by whole cell data without knowledge of the target. And this program went all the way to phase 2 clinical trials. So that was really the sort of early H3D success. We also have TB biology drug discovery programs, and obviously we've built a nice TB biology team within the IBM, really capitalizing on the world-class TB research that is done in this institute. And then the most recent addition to the portfolio is antimicrobial resistance, really with focus on gram-negative bacteria. And then the next ones are more technology platforms. So as I said, we've built some platforms, which I'll tell you a little bit more about, to really support drug discovery and be able to do this in an integrated sense. And then finally, we are heavily embedded in the academic environment. So we closely interact with Kelly Chibali's academic group, as well as other students within the IDM. And this is really great because drug discovery is obviously often on very tight timelines. And obviously, there's a lot of interesting science that you want to explore on either side. So sometimes projects transition from the academic group into H3D because they look like they're ready for prime time drug discovery. And other times it goes the other way, where it's stopped for some reason at H3D, but there's actually interesting biology or questions that could be answered and addressed with less of the time pressure or funding deliverables in an academic setting. Okay, so H3D really started with a very small team of medicinal chemists, and basically everything else was done with international partners, so biology, DMPK, that type of thing. Gradually, over the years, we've really been able to develop. We've been able to implement a lot of what's required in-house. So now we are fully integrated center. So here you'll see malaria biology, TB biology, AMR biology, and then what I've really been involved in is setting up what we call enzymology platform. So maybe we'll move away from enzymes at some point. So protein chemistry, and this has really enabled target-based drug discovery, where most of the things were done phenotypically before. And one of the things we've dabbled in, and I've been knowing Trevor for a long time, and we've always spoken about doing structural biology, but I was kind of a one-man show initially, and this is something we've just never really done in-house to any degree. But now I have a larger team, and this is really what the next part of the biology platform, what I would like to see in the coming years. And and then we have a very extensive DMPK or pharmacology platform, and this is really for supporting drug discovery. Obviously, you're looking for potency against the bug and your target, but really drug-like properties, understanding what happens with regards to metabolism is really key. And this is really underpinned by data storage and sample management, which becomes crucial for these kind of things. And over the years since 2010, a huge amount of data has been generated, and now we are using this data for AI and machine learning models to really try to streamline this. So this is quite a new and exciting addition to what we're doing. So a little bit more about the enzymology platform. So we do recombinant expression in E. coli and also insect cells. We work on plasmodium proteins, which are notoriously difficult to pretty much do anything with. They're very disordered, but we've managed to purify and develop assays for some challenging proteins. We do multi-step protein purification and then biochemical assay development. And what I learned quickly as a postdoc when I first started out and set up the first enzyme assay for PI4K, which was H3D's sort of flagship target. And I told the chemists I have an assay. And the next week it was like, how many compounds can you test? And I was there with my single channel pipette, my really expensive reagents. And I was like, maybe one, So a big part of what I've done since then is being able to really miniaturize the assays, cost effectiveness, semi-automate them, get things in place that we can actually profile hundreds of compounds instead of 3. So that's been something that I had to learn. And then we also do more detailed mode of inhibition studies. And now I have work in teams where chemists actually know what KM is and want to know more details about it because it becomes important for selectivity and that type of thing. So that's been a nice thing to see. And then really this information gets integrated into the project team, so the medicinal chemists can optimize the drugs. We do have structural-based drug design, and this relies on either structures from collaborators, typically, or published structures available in the PDB homology models in some cases where there's no structures. And again, always linking back to the disease biology. And then we have quite a lot of molecular biology capabilities in-house and also through collaborators where we can really show that our compounds are on target, because just because they inhibit that in the enzyme assay doesn't mean that that's the primary mode of action. So this is really a nice integrated thing. With drug discovery, projects are stopping and starting all the time. And so one of the key things is what do you invest resources in? So when often a project starts as a phenotypic program and then you identify a target, and now, the teams want target-based data. And the question is, can you develop your assay before the project stops for some reason? And so just knowing where to invest resources and the chemistry moves very quickly. We have very good chemistry labs. And so how do you decide, which targets are good enough to really invest in if it's going to be challenging to express purified protein? And then that extends to structural biology. I think we've had interesting projects, but often you really don't want the structures at the end of the project. You want the structures near the beginning, so that they can actually inform chemistry. So that's one of the challenges from my side is being able to keep up with our very prolific chemists. And so just some of the current targets. So I've done a lot of work on plasmodium kinases. This has been where most of my focus has been. We're also working on TB casing casinolytic protease, or the CLIP protease. And that's something Zyda has been working on in Pink Jersey. So you can ask about that. Then also on our AMR program, we're interested in outer membrane machinery. Getting into the bug is really key in gram negatives. And so if you can target things in the outer membrane, then that's a lot easier. Okay, so I'm going to go through this quickly. So in terms of plasmodium kinases, there have been several kinases that have been identified and validated as essential for plasmodium life cycle and have the potential to deliver antimalarials that are effective against multiple stages of the parasite life cycle. And the key for this would be for treatment, so blood stage, but also for transmission blocking and prophylactic treatment. So this is kind of the advantage of a target that is essential across the life cycle. So these are just 3 of them. So PI4K is actually the target of what I was referring to as the first successful phenotypic program that we ran. PI4K, this kinase, was identified as the target of that molecule. And so subsequently to that, we've had a number of backup programs also targeting PI4K. It's a highly effective target. The challenge is it's a kinase, it's a lipid kinase, and selectivity becomes really crucial. And so what's been nice to see is for some of the backup programs, now that we have the enzyme assay and the modeling, there's no structure for this kinase, despite lots of attempts to engineer it. It's it's floppy, it's got loops, and the structure hasn't been solved. But we use homology models, and we've been able to really design in selectivity for compounds, which has been nice to see. And then these are two protein kinases, similar, also essential for multiple stages. And we have these assays in house as well, and drug discovery programs ongoing for all three of these targets. So as I said, kinase inhibitors typically bind to the ATP binding site. And so really getting potent compounds, getting efficacy is not the problem here. Really selectivity and potential for off-target toxicity is really key, and really a big challenge. There are very big differences. There's between human and malarial kinomes, but obviously ATP binding sites ATP. So there is this challenge. But as I said, we've been able to use structure guided medicinal chemistry optimization to design in selectivity in some cases. Some of the other approaches that we've been looking at is targeted covalent inhibitors. So this has the potential to improve selectivity, but also crucially reduce the risk of resistance. So this is just briefly showing, this is PI4K, this is a ATP competitive inhibitor, and this is a cysteine that's actually not present in the human ortholog. So by adding a warhead here, we've been able to show covalent modification of this residue. So this just shows time-dependent shift in IC50 for the wild-type enzyme, whereas when you mutate the cysteine to alanine, you don't see that. So kind of showing that you're getting covalent binding. We've also been able to show this covalent bond formation through mass spectrometry. And then finally, mapping allosteric binding sites. So this is a new project that we're working with the Sanger Institute to try and identify other binding sites that we could target, which might give us some advantages in terms of selectivity. And then finally, with kinase inhibitors, you always get polypharmacology. So often they, hit multiple kinases, and selectivity is an issue, but because resistance risk is a really key thing for malaria, idea of being able to target two kinases at once can greatly reduce resistance risk. Okay, so the other target we're working on is the CLIP protease. So this is a multi-subunit protein. It's made up of this proteolytic core, and then it has this ATPase-dependent chaperone, which is responsible for unfolding the protein. So we have 3 biochemical assays for this protein. The one is the full complex assay, where you can degrade a protein, basically. The second one is just the peptidase assay. So if you don't have this unfoldase or ATPase chaperone, basically the complex can only degrade small peptides. And then we have the ATPase assay, which is just for the CLIP-C1 part of the protein. So this enables you to work out, you could have very different modes of inhibition for this target. And by doing all 3 assays, you can identify how is your inhibitor binding. And then coupled with this, we have our TB biology team who has developed some reporter strains and hypermorphs to also help understand on-target activity for this target. So we have currently kind of 2 programs looking at this target. The one is in collaboration with EU OpenScreen, where we've transferred our full complex assay. When I say we, I actually mean Zyda. He's transferred the full complex assay to collaborators in Poland, and basically they are screening this large compound library in a high throughput manner against this target. They've done the pilot screen already, and the data is looking promising. And hopefully we will find some interesting new chemical starting points. And then when we have them, we'll put them through all these assays to try to work out where are they likely to be binding. And then finally, we have another program that's ongoing. It's a formal hit assessment stage where this is in collaboration with the TB drug accelerator. And this is based actually on a published crystal structure where there's this allosteric binding site has been identified. So we constantly testing compounds for this program, which is an allosteric inhibitor program. And then finally, on the AMR side, we're interested in, as I said, these outer membrane targets. And as you can see from this diagram, just very briefly, there's a lot of machinery or complexes on the membrane that is responsible for keeping the integrity of the outer membrane, which really is really key for keeping our drugs out, right? And so we have some programs where they really phenotypic programs. We're not actively doing biochemical assays or work, but through our molecular biology team, we've identified resistant mutations in some of these components. And this is, really something that I see in the future we could benefit from some kind of cryo-EM, because obviously these complexes are not trivial to get structures of. Also, it's quite difficult to link these resistant mutations to direct target engagement. Are they resistant mechanisms, or is it really the target? And so at the moment, this project is really moving very fast and phenotypically driven. But I think there's some interesting stuff that we could do more from the biochemical and structural biology side. But again, it's that tricky balance of resources and where it's put on for a drug discovery program. So yeah, and I don't have an acknowledgement slide because basically it would just be way too many people. But this is just a photo of the H3D. It's a bit old, but the H3D team. So I'm happy to take questions.